Hi everyone, and welcome to our video on autosomal linkage. What we're going to be doing in this video is carrying on with our work on module 6, looking at these inheritance diagrams, and we're going to look at two parts of the specification: 6.1.2b part 1, the genetic diagrams to show patterns of inheritance, with that focus obviously on autosomal linkage, and 6.1.2b part 2, the use of phenotypic ratios to identify this autosomal linkage, including explanations for it. As always, the first thing we're going to do is look at this definition, because if we don't understand the term, how on earth are we going to get any further? So when we are talking autosomal linkage, we are talking about the fact that the gene loci present on the same autosome are often inherited together. So the genes are linked in how they're going to be inherited. Remember, an autosome is one of the 22 in humans non-sex chromosomes that exist, and therefore what we're really talking about here are that any of those genes located on one of the autosomes, so on the 22 non-sex chromosomes, will in some cases be inherited together. Okay, we're going to have a look at two different examples then. Okay, so the first one is if no crossing over has occurred. So if you remember, crossing over is something that happens as part of meiosis in prophase 1. So if we've got this scenario where there isn't crossing over, then all of the genes are going to be inherited as a single unit on that particular chromatid. So what we've got then is, on the little diagram to the left here, we can see we've got our two homologous autosomes here. So the blue one, we've got both capitals. So on each chromatid you can see there's a capital A and a capital B. And then on the red ones we can see there's lowercase a and lowercase b. So when it actually splits into the individual chromatids, then we end up with two blue ones with a capital A and a capital B on each, and two red ones with a lowercase a and a lowercase b. If we have a look at this as an example then, because again, it's easier sometimes with genetics to look at examples to be able to visualize what's happening. What we're going to have a little look at in the world of plants is flower color and pollen shape. Now, the genes for flower color and pollen shape are located on that same chromosome. And flowers will either be purple or red. So purple is given the capital P and red the lowercase p. And the pollen grains will either be long, capital L, or short, lowercase l. So they're the options: purple or red flowers, long or short pollen grains. Both of the genes on the same autosome. So we could end up with a little question like this: work out the F2 genotype and phenotype for crossbreeding two heterozygous individuals of the F1 generation. So what we're told in the question, they're heterozygous individuals first of all. Now, if they're heterozygous, this means that they must therefore have one purple and one of the red flowers, and one long and one short pollen grain gene. Because these are linked, we need to write this in a slightly different way. So what we actually have, you can see how I've set it out in this little row here. We put them basically on top of each other, like a little stack of genes, if you like. So we're going to have one capital and one lowercase of each of the P's and the L's. They're both heterozygous, so they're both going to be purple long-grained parent plants, because purple is dominant and long is dominant. Therefore, what we've got, capital P, lowercase p, capital L, lowercase l. And obviously we're crossing them, so that means we end up with that same situation for both parents. When we then split those into the individual gametes, what we end up with is that we've got to split them as those vertical columns. So where we've got the two capitals, they must go into one gamete. Then you've got the two lowercase have to go into the other gamete. So what we can see on that third row are the potential gametes. We've got one both capitals and one both lowercase for each of the parents. We then need to work out the potential genotypes of the offspring, and again, they've got to stay together. They are linked, remember. So we get one of our two capitals of parent 1 going straight down, and then we get one of our gametes from the other parent coming across. So you can see how this is all worked out. Put the different colours on those lines to hopefully make it easier to see. So when we then look at the potential phenotypes that we get, this first one that we have on the far left hand side, we've got two capital P's, two capital L's. That's going to be a purple, long-grained plant. The next one along has one capital and one lowercase for each of those characteristics. Therefore, it's going to be purple and long-grained again. Same for the third one of each, therefore purple and long-grained. And then finally, we've got two lowercase p's, two lowercase l's, therefore it's going to be red and short-grained pollen. So we actually end up with this final ratio of 3 purple long-grained plants to 1 red short-grained plant. Now, what it actually constitutes is 1 homozygous dominant, which is the one on the left. We then have 2 of the heterozygous, which are these two, and then 1 homozygous recessive. So it's a 1 to 2 to 1 ratio there. What we then can say is that if this F2 generation has a 1 to 2 to 1 ratio, we know this is autosomal linkage, because as we saw earlier on when we did our dihybrid inheritance, if we've got these two genes that are not linked, that are inherited independently of one another, then the ratio we would expect to see is 9 to 3 to 3 to 1. But we don't have that. We've got 1 to 2 to 1. So because we don't have that expected 9 to 3 to 3 to 1 ratio, we know that something else is happening. And the something else is our autosomal linkage because of that 1 to 2 to 1 ratio. The second scenario that we see with autosomal linkage is when crossing over does occur in prophase 1 of meiosis. Now, if we think back to meiosis, hopefully we remember that crossing over is where we are exchanging alleles between those two sister chromatids, and as a result of this we're getting this reshuffling of the alleles. What we end up forming is what we term a recombinant gamete. So if we have a look at the left, we've got the same starting points as we did previously. We have a blue chromosome with a capital A and a capital B on each chromatid, and we've got a red chromosome with lowercase a and lowercase b on each chromatid. Now when crossing over occurs, we can see this little region here, we're swapping two parts of the chromosome over. So basically what we're seeing is two bits of chromatid are being exchanged with one another. This then results in our 4 gametes, 2 of which are now recombinant. We still have one blue with a capital A and capital B, we still have one red with lowercase a and lowercase b, and then these two in the middle are going to be the recombinant gametes, because if we look, they've got a section of the blue with the capital on it and a section of the red with the lowercase. So what we've done through this process of crossing over is we've created a new assortment of those alleles to what we would have had without the crossing over. Now, because this crossing over has taken place and we've reshuffled those alleles into new combinations, then we can actually end up with what we term unexpected phenotypes in our F2 generation. So what we obviously would have said previously, if we started off with those same 2 chromosomes, they then split into those 4 gametes without the crossing over, then we ended up with obviously just those 2 traits. They were purple long-grained or red short-grained plants. That was it, and it was in a 3 to 1 ratio overall. So the 1 to 2 to 1, that was obviously 3 purple long-grained to 1 red short-grained. If we've got this crossing over occurring, then we're not going to see that because we've now got new combinations of alleles to be inherited. So we're going to get some unexpected phenotypes cropping up. We're also not going to get our unlinked ratio of 9 to 3 to 3 to 1. So because we've now turned out a different ratio than the standard 3:1 that we would expect if it was autosomally linked without crossing over, and it's not the 9:3:3:1 ratio of unlinked genes being inherited at the same time, then what we can say is that these are autosomally linked and crossing over has occurred. One last point that we should really just make before we leave this topic is that when we're thinking about the likelihood of recombinant gametes forming, we need to think about the position of those individual gene loci on the chromosome. Because if we're thinking about two loci that are far apart on the chromosome, opposite ends for example, then what we have is a much greater chance of recombinant gametes forming. If we've got these two genes being located very close together, then it's less likely that we're going to get any reassortment. Because if you imagine the scenario where we've got our little genes located in that same region, that whole section could be swapped over. Whereas if we've got our genes located at opposite ends of that chromatid, we'd only be swapping one of them at a time. So we get that reassortment, that shuffling. So basically, the closer together the genes are, the less likely they are to form recombinant gametes. The further apart they are, the more likely they are to form recombinant gametes. As always, I hope you found this video useful, and don't forget to subscribe to the channel so you can see when we next upload another video, and of course head on over to the website where you can find an array of other A-level biology resources to help you in your studies.